Hello, AP Biology students. We're going to go over the fourth section in Unit 5. And in this section, we're going to cover dihybrid Punnett squares. Remember, up until this point, the content has been focused on monohybrid Punnett squares and what Mendel saw with his experiments. During his experiments, Mendel started to wonder what would happen if he studied two different traits when looking at his data. So basically, he wanted to see if there was another pattern that he could see when looking at these two traits. And he also wanted to see if there was some influence the traits had on each other when looking at the offspring. One of the things we're going to touch upon in this section, in the next section, is something called a locus. The locus is just the location of a gene on a pair of homologous chromosomes. You can see it in the pair of homologous chromosomes here. The gene location is just the gene locus. This is again just where it's physically located on these pairs of homologous chromosomes. That's going to be really important when looking at the next section. When looking at a dihybrid cross, we're looking at the cross between two traits. Remember that gametes are going to have only one of each of the alleles in a genotype. However, when we're looking at two different genotypes, we need to make sure we account for every possibility the gamete may have. When looking at big A, big B, little b, we have four different combinations. You can see what those combinations are here. This is because through meiosis, each of these gametes is possible through independent assortment and crossing over. Those possibilities are going to be placed again on the tops here and on the sides of a Punnett square. You can see our Punnett square is much larger. Our monohybrid Punnett squares only had 4 possibilities, where a dihybrid Punnett square is going to have 16 possibilities. And just like we did with the monohybrid Punnett squares, each of these gamete possibilities at the tops and the sides are going to be placed in each one of these boxes going down and going across. Let's take a look at pea plant color and the surface texture of a pea plant pod. In this example, yellow peas are going to be dominant to green peas, and in this example, smooth is going to be dominant to wrinkled. We're going to cross two heterozygous parents in this example. You can see they are both going to be big A, big B, little b. So we're going to place their possible gametes like we showed you before at the top here and on the sides. Remember, just like we did in the monohybrid Punnett squares, we're going to place each of these alleles in each box below and each box to the side. So for this first box, you're going to combine big A, big B with big A, big B. And this first box is going to be big A, big A, big B, big B. Whenever we combine the different gametes, we again place the A's together and the B's together. For this second box, it's going to be big A, big B, little b. The third box is going to be big A, big B, big B. And for this last box, it's going to be big A, big B, little b. Here are all of the combinations or possible outcomes, possible zygotes for this dihybrid cross. What this dihybrid cross allows us to do is to show what the possibilities are of the offspring. Again, it's not going to say that we have to make 16 different offspring from the combinations or the cross of these two parents. This dihybrid Punnett square is just showing us the possibilities of the outcomes of those zygotes. If it makes a little bit more sense, you can see what those possibilities are going to look phenotypically. And what this allows us to do is to predict the outcomes of the offspring. If you look here, yellow and smooth have the most genotypes, and they're going to have the highest ratio, 9 out of 16, or about 56%. Yellow and wrinkled only have 2 different genotypes, and they're going to count for 3 out of the 16 different offspring, which is about 19%. Same with the green and smooth, 2 different outcomes, 3 out of 16, about 19%. And the least likely is green and wrinkled. It's because they only have 1 genotype. They only account for 1 out of 16, which is about 6%.